Inside a sea anemone, you can find a family whose future depends on one fish transforming after a death. The largest resident is female. The second largest is a breeding male, and smaller fish wait below them. When the female disappears, the social order does not simply replace her with another arriving adult. The breeding male begins changing sex while the largest subordinate rises to become the new male. This sequence keeps reproduction possible within a refuge that fish cannot safely abandon without consequence. The familiar orange clownfish, therefore, carries one of reef biology's most dramatic solutions to partner loss. Transformation is real, documented, and governed by anatomy, hormones, behavior, and social rank. It is not an instant switch, and it does not occur because the fish consciously chooses survival. The change unfolds through physiological responses after the dominant female's controlling presence has vanished. Each stage answers a specific problem created by life in isolated partnerships among dangerous reef spaces. Leaving to search widely for a female would expose a small fish to predators and uncertain shelter. Remaining without a breeding female would eventually end that resident group's reproductive contribution. Sequential hermaphroditism provides another route, rebuilding a breeding pair from fish already inside the anemone. The mystery is how a social absence becomes a biological signal capable of reorganizing an adult body. Its answer begins with the stable hierarchy visible before any transformation starts. Clownfishes belong to the subfamily Amphiprioninae and live in close association with particular sea anemones. The common clownfish, Amphiprion ocellaris, offers a well-studied example of male-to-female sex change. Every individual develops first along the male pathway, although social position determines whether it breeds. This pattern is called protandry, meaning male function precedes possible female function later in life. A resident group usually contains one breeding female, one breeding male, and several smaller non-breeders. Size differences are maintained through dominance, submission, and restricted growth among lower-ranked fish. The female asserts rank through interactions, and the male directs similar pressure towards subordinates. Smaller members often perform appeasing movements that reduce conflict and preserve their place in the group. This hierarchy is not merely visual order because it regulates access to reproduction. The anemone provides shelter among stinging tentacles that deter many potential predators. Clownfish mucus and behavior help them live among those tentacles without receiving ordinary damaging stings. In return, their presence can benefit the anemone through nutrient delivery, water movement, and defensive interactions. The partnership creates a valuable fixed address, making departure potentially more costly than transformation. Breeding pairs prepare a firm surface near the anemone before eggs are deposited. The female lays adhesive eggs, and the male fertilizes them externally during spawning. Afterward, the male commonly fans and tends the developing clutch, removing compromised eggs. These actions establish functioning sex roles before the hierarchy is disturbed. Male and female clownfish possess reproductive anatomy differentiated for their current roles. Sex change requires coordinated remodeling rather than a superficial alteration of color or behavior. The gonads transform as ovarian tissue develops and testicular function recedes. Hormonal pathways shift alongside gene activity, though exact timing varies with species and conditions. Mainstream interpretation identifies removal of social inhibition as the crucial trigger. The verified sequence is clearest when the dominant female is experimentally or naturally removed. Within a short period, the former breeding male begins behaving as the group's dominant fish. Aggressive and territorial interactions change before complete reproductive anatomy can be rebuilt. The largest subordinate then moves upward, gaining the behavioral position previously held by the male. If you returned immediately, social roles might look altered, while internal transformation remained incomplete. Over subsequent weeks, gonadal and hormonal changes can establish functional female reproduction in the former male. Exact schedules should be reported cautiously because temperature, species, age, and study design affect progression. The replacement male matures into reproductive function while maintaining a smaller body size. Eventually, the reorganized pair can produce and care for another clutch of eggs. It is the disappearance of the top-ranked female within a recognizable social structure. Visual, chemical, and behavioral signals may all contribute, but their relative roles remain under investigation. Experiments that alter group composition allow scientists to test which social interactions suppress transformation. Hormone measurements reveal associated physiological changes, while tissue examination confirms gonadal remodeling. Gene expression studies can identify molecular pathways activated during the transition. These methods connect observed rank changes to internal biology without claiming access to the fish's experience. The evidence strongly supports social control, but a single master switch would oversimplify the process. Brain signaling, endocrine regulation, gonadal feedback, and behavior interact across different timescales. A dominant posture can emerge sooner than mature ovaries, showing that transformation has separable stages. Scientists continue testing how those stages remain coordinated and when reversal becomes impossible. In clownfish, the established direction is male to female rather than repeated switching in either direction. That limitation distinguishes protandry from other forms of sex change found among reef fishes. Some wrasses change from female to male, usually under different mating systems and social pressures. Those examples demonstrate multiple evolutionary routes, not one universal mechanism shared identically by all fishes. For Amphiprion ocellaris, the evidence belongs to a particular ecology centered on anemones and size hierarchy. Claims should not be extended automatically to every clownfish population without species-specific observation. Still, the core pattern has broad support from behavioral, anatomical, and endocrine research. Why would natural selection favor beginning as male and becoming female only after reaching dominant size? In clownfish, larger females can generally produce more eggs than smaller females. A small individual can function effectively as a male before gaining the size needed for high female fecundity. When the largest female dies, the next largest fish is already positioned to assume the egg-producing role. This size advantage model offers the mainstream evolutionary explanation for protandry. Describes differences in reproductive payoff, not a conscious calculation performed by individual fish. The cue of female loss activates an inherited flexible system shaped across many generations. The consequence is continuity within a scarce shelter where unrelated replacements may not arrive safely. For the subordinate, promotion also changes feeding access, growth, aggression, and reproductive opportunity. As social pressure relaxes, the new male can grow and participate in spawning. Hierarchy, therefore, links behavior to body development throughout the entire group. Larger outsider enters. Conflicts may rearrange rank and complicate the expected sequence. Local density, anemone distribution, and predator pressure influence how stable these groups remain. Warming, bleaching, and habitat degradation can damage anemones, removing the structure that makes this system possible. Fish capable of changing sex still cannot reproduce successfully without sufficient habitat, food, and partners. Aquarium popularity also creates human stakes when collection is poorly managed, though many clownfish are captive bred. Understanding social biology improves welfare by discouraging arbitrary grouping that produces chronic aggression. It also helps population models represent breeders accurately instead of counting every adult as equivalent. When researchers survey a reef, size and rank can reveal likely reproductive roles without destructive sampling. Yet visual inference must be checked because behavior alone cannot confirm fully functional anatomy. If you see two fish together, body size suggests hierarchy, but does not reveal every transitional stage. Future studies can track identified individuals from female loss through successful spawning over longer periods. Non-lethal hormone sampling may clarify timing while reducing disturbance to small resident groups. Controlled playback or model experiments could isolate which female signals maintain inhibition in the breeding male. Genomic and cellular work may reveal how adult gonads change identity while preserving healthy tissue function. Comparisons among Amphiprion species could test whether ecological differences alter speed, thresholds, or social sensitivity. Field studies are essential because laboratory groups cannot reproduce every risk imposed by open reefs. Researchers also need observations during anemone decline, when shelter loss may interrupt ordinary succession. The strongest future evidence would connect a precisely measured social cue to brain signaling and completed fertility. Until then, the pathway is well supported in outline, but incomplete in its finest mechanisms. You should picture transformation as a coordinated process, not a sudden exchange of one body for another. Behavior changes first, internal tissues remodel over time, and successful eggs provide the functional test. The family persists because an existing resident can fill the missing reproductive role without crossing exposed reef. That flexibility increases reproductive continuity, although it cannot guarantee individual survival or population recovery. The opening anemone remains occupied, but its hierarchy has been rebuilt around a changed adult. What survives is not a fixed identity, but a breeding system adapted to loss within a dangerous reef. The fish that becomes female demonstrates a verified biological transition whose social trigger is clear, even while molecular details remain unresolved.